Hello, my lovely anatomist and physiologist, Michele Glass here, and welcome back to another Chapter 3 video. This video is all about how proteins are made. So let's take just a few minutes and go back to Chapter 2 content and remind ourselves about the significance of proteins. What are some examples of what proteins do for the cell, for the body? So remember from Chapter 2 that proteins act as buffers. Buffers, remember, help to regulate pH, right? So it helps to prevent large fluctuations in pH. Proteins are important for cellular structure. We just got done talking about the cytoskeleton. We talked about how the cytoskeleton is involved in movement that's happening within the cell and also movement of the cell itself. We studied tissues in our lab component, and we saw the importance of collagen and elastic fibers in some of our connective tissues, especially. Proteins are important as enzymes. And remember, enzymes are those proteins that catalyze or speed up chemical reactions. And remember, essentially in humans and our cells, you should consider no reaction is taking place without its corresponding enzyme. So production of enzymes is basically a way that the cell controls what kind of chemical reactions are happening in that cell. So this involves a lot of regulation. Proteins are key in our immune response. So antibodies are proteins. And we also talked about those glycoproteins as part of our glycocalyx, and that was involved in our cell recognition. So part of our ability of cells to recognize which cells belong in your body and which don't. And along those same lines, we can talk about the cell membrane channels. So this is going to be critical for transport across the membrane. We had cell membrane carrier proteins. Again, critical in transport of material across the cell membrane. We'll see also a lot of our cell receptors are proteins. We see also that our neurotransmitters, which are our communicating chemical for nervous system, are composed of proteins. We have hormones, which is another important chemical messenger. We're often composed of hormones. So we're seeing, this huge range of jobs for these molecules. And so it makes sense then that we should spend a whole little section here talking about how proteins are made. And when we talk about how proteins are made, remember that they're being coded for by that molecule called DNA, or deoxyribose nucleic acid. And so let's go back and talk about DNA for just a little bit. So deoxyribose nucleic acid is our abbreviation for, is our full name for the abbreviated DNA that we see quite a lot. Remember that deoxyribose nucleic acid is going to be housed inside the nucleus of the cell, the cellular nucleus. And you should consider that this molecule is always and forever kind of like held secure in this structure, in this organelle, except when the cell is doing cell division. So all the rest of the time, it's only and ever going to be found inside the nucleus. And so let's go back just a little bit and remind ourselves about our nucleus so we can draw out for ourselves. Kind of our nucleus is going to have a double membrane, and then it's going to have pores in that nuclear envelope. And so our DNA molecule then basically is housed inside our nucleus here. So we'll say this is our nucleus. And DNA is not able to fit through those pores. It's going to be too big of a molecule. When we are talking about our DNA molecule, we'll see that it can come in two forms. It can come in what is called chromatin, which is like our relaxed DNA molecule. I like to think about this as, you know, when you are living in a space, you have maybe clothes hung up in the closet, put away maybe in the dresser, you have pictures on the wall and knickknacks out on your furniture. You maybe have a desk lamp and you have your bed spread out, and maybe you have a book rack, or maybe you have your movies, or basically all of your stuff, whatever is your stuff, you have it just like out in the room where it's easily able for you to access it and use it. I like to think about the chromatin kind of like that. So the chromatin is the DNA molecule that's open and relaxed. It's easy to access in order to do the process of making proteins. It's in this stage of chromatin when the cell can duplicate or replicate that DNA molecule. So the chromatin is the relaxed, like I'm going to call it functional form of the molecule. When we're getting ready to move to a new room or move to, you know, a new apartment or new house, new place, whatever it is, you know, very often if we're able to, we're going to pack up our belongings, right? So we're going to pack up our clothes. We're going to pack up those knickknacks. Maybe if we have breakable items, we wrap them up in a way to keep them secure. We put things in different boxes or suitcases or bags, you know, whatever we can do to kind of pack up our stuff. And we do that so that we don't lose anything when we move it. We do that so that we don't break stuff when we move it, right? And so we have a form of DNA that's kind of like that same thing. So we have what's called chromosome, which I'm going to call this like the packaged form. And we see this during cell division. And it's not functional at this time. We're not able to go in and use it to make proteins. We're not able to go in and replicate it. This is just like the packaged up form of DNA. So we'll talk about that as we go through in just a little bit. Now, reminding ourselves about DNA structure, we can, if we want, remember, construct like a ladder. So we're going to have two vertical lines, and then we're going to have our horizontal ladder rungs kind of connecting those two vertical lines together. So we do talk about this as being a double-stranded molecule. And those two vertical lines, those two like sides of the ladder, we would consider to be a sugar phosphate backbone. Which, remember, in the case of deoxyribose, that OSC ending, remember, clues us in that this is our sugar in this particular molecule. And then those horizontal lines, or those rungs of the ladder, are going to be our nitrogenous bases. And remember, this can be adenine, which we can abbreviate with an A. And adenine always pairs with thymine, which we can abbreviate with the T. And we can remember apples grow on trees to help us remember that adenine pairs with thymine. And then we have the nitrogenous base cytosine, which can pair with guanine. And we can use the letter C to abbreviate cytosine and the letter G to abbreviate guanine. And so apples grow on trees reminds us that A pairs with T, and the cars in the garage helps us to remember that the cytosine pairs with guanine. And so what we're seeing is across one strand, we have A and C and so forth. And on the other strand, we have the T and G and so forth, right? And if I wanted to, I could just fill in the rest of my ladder, and I'm just going to randomly do this. So on one side of my strand, I'm going to have A, C, A, T, C. And on the right hand side, pairing with that, I'm going to have T, G, T, T, A, G. Now what we'll see is during what we're going to call interphase, and we're going to talk more about this in the next video, we're going to get DNA replication. And the way that the DNA is replicated, it's going to involve some important enzymes. And we don't have to know a whole lot of details, but we'll mention helicase as an enzyme that's going to unwind that double strand. That's the other piece. If we go back to our picture of our ladder, we then have to like hold on to the top and the bottom and then twist. So you make a spiral, right? So we can kind of spiral around to sort of show the helix. So we can go up here and say that it's also a helix. The helicase, then ASE ending, is reminding us it's an enzyme, and the hela part refers to helicase. So the helicase enzyme is going to unwind or untwist that. And then we have a molecule called DNA polymerase, which is going to build the new strand. So I'm going to just unwind the strand that I wrote. So I'm going to do on my left side, the left side of my ladder. So I'm going to have my A, C, A, T, C. And I'm going to give quite a lot of space. And then on the right-hand side, I'm going to write out my right-hand side of my ladder. So I'm going to have T, G, T, T, A, G. And then I'm going to actually change my color to represent my DNA polymerase coming in and replicating. So where I had my left side and I have my ACAATC, I'm going to keep that in black. And then right beside it, I'm going to draw a complementary base pairing in blue. So where I have A, I'm going to pair it with T, and where I have C, I'm going to pair it with G, and so forth. So I'm going to build out that strand. And then I'm going to go to my black strand on the right where I have TG, TTAG, and I'm going to build based on the rules of complementary base pairing, so that now I have two identical strands. They look the same like each other. They look like the original strand that I drew. And so we can talk about this as being semi-conservative replication because I've kept an old strand paired with a new strand. Okay, and this process of DNA replication is happening inside the nucleus. This is happening specifically during a phase of the cell cycle called interphase, which we'll talk more about in just a little bit. So that's our quick review of our DNA molecule. Let's talk for just a minute about RNA molecule and how RNA molecule is different. Now, RNA molecule, RNA stands for ribose nucleic acid, and we see ribose has that OSE ending, and so we know that that means that we're talking there about a sugar, right? And so here is one of our key differences between DNA and RNA. So you could remind yourself, you could draw a little table where you have DNA versus RNA, and DNA has deoxyribose, and RNA has ribose as the sugar. Now, a ribose nucleic acid is going to look like one side of the ladder. So another key difference is that RNA is single-stranded, whereas DNA is double-stranded. And it's still got a helix, so instead of just drawing like a straight line down, I'm going to actually make it look sort of twisted like a ribbon. And we still get a sugar phosphate backbone, but now remember, we've already said our sugar here is ribose instead of deoxyribose. And then sticking off of our like a ribbon, we still get those nitrogenous bases, but here we're going to have adenine, we're going to have uracil, we're going to have guanine, and we're going to have cytosine. And so I'm going to use A to abbreviate adenine, and I went ahead and wrote out uracil, which you can abbreviate with the U, and then I have my G for guanine and my C for cytosine. So when we look at our nitrogenous bases in DNA, we have A and T, and we have C and G. And when we look at RNA, we have A pairing with U, and we have C, and we have G. So that's going to be an important difference. When we talk about RNA, we're going to see there's 3 different types. So we're going to have what's called messenger RNA. We're going to have what's called ribosomal RNA, and we're going to have what's called transfer RNA. And what we'll see is we'll abbreviate messenger RNA with a little m and then RNA. And we'll use a little r and then capital RNA for ribosomal RNA. And we'll use a lowercase t and then a capital RNA for tRNA. Okay, and so it'll look like lowercase m, capital big RNA, lowercase r, capital RNA, lowercase t, capital RNA. Okay. Now, as we look at these, they each have their own special jobs. And so messenger RNA is going to take a copy of what we call the gene to the ribosome. So we'll talk more about this as we go through the process. And then we're going to see that the ribosomal RNA actually forms the ribosome. Now, ribosome is a structure that we talked about earlier. Remember, ribosome is actually the location of protein synthesis. And let's also go back to our previous drawing of the nucleus, and let's draw kind of a dark circly structure inside called the nucleolus. And this is going to be where lowercase r, capital RNA is produced. So it's the darkest structure inside the whole nucleus. Okay. And then when we talk about the tRNA, the tRNA, is going to transfer, it's going to transfer amino acids to the ribosome. I like to use this analogy of cooking, right? And so the messenger RNA is like the recipe. The ribosomal RNA serves as like the kitchen. And then the transfer RNA is going to bring ingredients. Where remember, the amino acid is the building block or the monomer for proteins. It's part of that basic primary structure of a protein. Okay,